Dear students, today we are going to see the third unit of our syllabus, the cell structure and its functions. So in the particular unit we are going to cover three major areas, that are the cell as a unit of life, the biomolecules, and cell cycle and its cell divisions. Okay, and first we are going to start with the cell as a unit of life. Okay, so before we are entering into the details of the different types of cells and its functions, first you should know something about what is cell. Okay, so as we all know, the atoms are the basics of chemistry, the cells are the basics of the biology. Okay, so without cells you cannot explain anything about the living organisms or biological entities. Okay, so the point you have to remember over here, all the living cells or all the living organisms are made up of the basic unit called cells. Okay, so the cells are the basic units of biology, or the cells are the basic units of any living organisms present all over the world. Okay, so the next point you have to remember over here, the origin of the cell theory or the history behind the cells. And the history starts with the man Anton von Leeuwenhoek. Okay, so the man who has discovered the microscopes. The preliminary biological microscope have been discovered by the person called Anton von Leeuwenhoek, and he has observed the first living cells under the microscopes. Okay, so the history of cells begins with the history of discovery of microscope by the person called Anton von Leeuwenhoek, and the next major scientist you have to remember while you are studying the cells, Robert Brown. Okay, and he has discovered the presence of structures called nucleus. Okay, so these are the two major points you have to remember while we are discussing about the history. Okay, the first thing, the discovery of microscope. There are so many microscopes present in those times. The microscope used for astro studies. Okay, but the point you have to remember, the biological microscope have been discovered by the Anton von Leeuwenhoek, and he was the first person observe the living cells under the microscope, and Robert Brown who has find out the structures called nucleus present in the particular cell structures. Okay, and next point you have to remember in the introduction, the microscopes. So as we all know, there are two major classes of microscopes, that is light and electron microscopes. Okay, so what is the differences between these two microscopes? The light microscopes, they are going to use the photons or the light source as a energy for the illumination, and the electron microscopes, they are going to use the electron beams. Okay, so the point you have to remember over here, the light microscopes, they are going to use the photons or energy. Photons are the particular light sources or energy sources to illuminate the particular slides, and the electron microscope, they are going to use the electron beams. So we all know how to perform the staining procedure. So we will prepare a smear on a glass slide and we will observe it under the microscope. Before that we will stain it with some particular charged staining solutions. So what will happen over here, those charged stains will stain the particular internal and external structures of the particular microscopic cells, and after that, after the drying, while you are observing it under the microscope, you can see the morphology of the internal and external structures stained with the particular stain. Okay, so what will happen while you are using the light microscopes, the light energy or the photons will illuminate the particular stained smear, so that you will see the external and internal morphology of the particular cells. In case of electron microscope, the electron beams we are going to use. So what is the differences between these two sources? Okay, so then you have to remember another term, that is called resolving power. So what is the resolving power? The resolving power, so I will explain the resolving power. So there are 2 points I have placed over here. Can you distinguish these 2 points? Okay. There are another 2 points I am placing it over here. Can you distinguish these 2 points? Can you hear? So for the naked eye we can easily distinguish these 2 points, and somewhat we can distinguish these 2 points. This is difficult, this is more difficult, and this is we cannot distinguish from the particular distance. So the point is, for a naked eye there is the resolving power at some point or at particular, okay, for a particular range it can resolve the nearest 2 points. Nearest 2 points. Okay, so that is what we are calling that resolving power. Okay, for a naked eye they can distinguish at a particular range, they can distinguish the nearest 2 points. So what we are going to do with the microscope, we are going to increase the resolving power of our eye. Suppose if you are going to use the light microscope, you are increasing your resolving power to some extent. At what extent? We all know the microscopic cell structure. The prokaryotic cells, it will be around 1 micrometer to 5 micrometer size. Okay, 5 micrometer size. So what it means? While you are observing it under the microscope, you can distinguish the microscopic structures in the size range of 1 to 5 micrometer cell size. Can you distinguish those size with the naked eye? You cannot. So what happens over here? The light microscope increases the resolving power of your naked eye to distinguish the 2 points in the size range of micrometer. Okay, so the electron microscopes are always better than the light microscope in case of resolving power. Why? The point you have to remember over here, they will increases your resolving power in the nanometer scales. That means the resolving power of the electron microscope are always better than the light microscope, so that you can easily distinguish the internal structures and external molecular structures of any cell in the cell structures. Okay, so the points you have to remember in the first introduction class, there are 3 things. The cells are the basic units of life, and the discovery began the cell's presence. The Antony van Leeuwenhoek and Robert Brown. Then the microscopes. There are 2 classes of microscope, light microscopes and electron microscopes. Then the point you have to remember in the microscopes, the resolving power, light energy or the photon energy is being used in case of light microscopes. So the resolving power of light microscopes are lower than the electron microscopes because they are going to use the electron beams as a energy source. Their resolving power are better than the light microscopes. Okay. And now we are going to enter in the topic, the cell as a unit of life, and we are going to start with the cell theory. And the cell theory explains why the cells are the basic units of the biological entities. Okay, so the cell theory starts with 2 major scientists, Theodor Schwann and Schleider. Okay, so in 1838 or 39, okay, in 1838 and 39 they have discovered the structures called the cells. Okay, so both of them studied the plant and animal tissues. So what are tissues? Okay, so the tissues are the structures, microscopic structures are all the living cells, all the multicellular organisms are made up of tissues. What are tissues? The tissues are group of cells. Tissues are a group of cells. The point you have to remember over here, while they are studying, the Schwann and Schleider studying, they didn't know something about the tissues or the cells. They are the first persons to conclude the presence of the cells. They have discovered the structures of cells or the cells are the unit of life in the tissues. Okay, so they have discovered that some box-like structures present in all over the tissues of the plants and animal. Okay, and they have named them as cells, and they have concluded as a cell theory. The first point of cell theory you have to remember over here, concluded by Schwann and Schleiden. All the organisms, that is a major word you have to remember. All the organisms or all the living things are made up of cells or its byproducts or its. Okay, so all the living organisms are made up of cells or its products. Okay, but this is not a complete cell theory, and the cell theory was completed by another scientist called Rudolf Virchow in 1855. So in 1855, Rudolf Virchow discovered the cell division. So what is cell division? The cells are formed from pre-existing cells. Okay, so all the living cells are formed from pre-existing cells. Okay, so with this the cell theory is getting completed. So there are 2 points you have to remember in case of cell theory. The first thing, all the living organisms are made up of cells and its products. And the second major point you have to remember over here, proposed by the Virchow, the cells are formed from the pre-existing cells by means of cell divisions. Okay, so these are the points you have to remember in case of the cell theory. Okay, so what and all we have completed till now? We have studied what is cell, and we have studied something about the history behind the cells. Then what is cell theory and who has proposed the cell theory, and what are the points you have to remember in case of cell theory. And now we are going to enter into the topic called the prokaryotic and eukaryotic cells.